All right, good morning, everyone. And thank you so much for joining us here today. Are you able to hear me and see me okay? Awesome. Well, my name is Ayla, and I am the Ocean Science Educator for Ocean First Institute. And happy Earth Day, everyone. We, Ocean First Institute, are an ocean conservation nonprofit that's based here in Boulder, Colorado. And we protect the ocean through both research and education and study animals so we can share how to better protect and understand the ocean. Today, it's so exciting to have so many people joining us from all over the world and country to learn whether a shark can really smell a drop of blood in the water from a mile away, and other amazing facts about one of sharks' most powerful senses. So we're going to have a presentation that will take about 20 minutes, and then we're going to take questions for the rest of the time for about 25 minutes. So during the presentation, if you have any questions, please do feel free to type those into the Q&A box, and our panelists will answer those at the end of the presentation. This is also being live streamed on Facebook, so if you have any friends or family who weren't able to get into the Zoom but want to still see it, send them over to the Ocean First Institute Facebook page. So now I'm so excited to welcome both our Executive Director, Dr. Mickey, and our speaker today, Dr. Trish Meredith. So Dr. Meredith is an Assistant Research Professor at Florida Atlantic University's College of Education and serves as the Director of Research for A.D. Henderson University School and FAU's High School. She holds a doctorate in integrative biology and focused her dissertation research on the sense of smell of sharks and their relatives. Her postdoctoral work at the University of Miami's Miller School of Medicine addressed the impact of fat hormones on our sense of sweet taste. As research director, Dr. Meredith has designed and implemented the FAUHS research program and serves as the principal investigator for the FAUHS OWLS Imaging Lab, which both supports dual-enrolled students and undergraduate research. Dr. Meredith continues to study the sense of smell of sharks and their relatives to learn how sensitive they are to odors and the reasoning behind the organization of their noses. I'm also very excited to be joined today by Dr. Mickey. She is the Executive Director of Ocean First Institute and one of the top shark experts in the world. She also holds a PhD in integrative biology and speaks, teaches, and conducts research around the world, including in South America, Africa, Asia, and Australia. Her work has been covered by the BBC, National Geographic, and even Discovery Channel's Shark Week. So I'm now going to turn it over to Dr. Meredith to tell us a little bit about sharks and their amazing senses. And like I said, feel free to ask questions as we go along. All right, Dr. Meredith, take it away.
Okay, thank you, everybody. All right, can you guys hear me and see me okay?
Perfect. Welcome, Trisha. Thank you.
Thank you so much. All right, so I'm going to talk to you today about the sense of smell of sharks and their relatives and get to this question that we hear a lot in shark documentaries. Can sharks smell a drop of blood a mile away? And what you see here twisting around on your screen is a nurse shark head. And the reason it looks a little bit strange is that it is a 3D X-ray produced by a micro CT scanner of this nurse shark. So you can see the rough skin, the jaw is really highlighted in the teeth. And up here, if you can see my cursor, this light gray area is where the nose is. So we're going to talk about that. Before I jump in to talk about sharks and their sense of smell, first, I want to kind of let you know how we're similar to sharks in the way that we smell things. So what we're talking about when we think about the sense of smell is being able to sense chemicals in our environment. So we're talking about chemical sensitivity. So this is one of our earliest receptor systems, and it occurs in all organisms, and even each of our cells inside of our body needs this ability to detect and respond to the chemicals in its environment. So we have a couple of senses that deal with chemicals, our sense of taste and our sense of smell, which is the one we're going to focus on today. The sense of smell works basically the same in most vertebrates. So I wanted to let you know that when I talk about the shark sense of smell, there are a lot of similarities with us and them. So the way this works is that odors, either floating around in the air or dissolved in the water, depending on where you live, they float into the nose through the nostrils and they enter an olfactory cavity. So this cavity inside of our nose, and inside that cavity, there's a sheet of skin on the roof of our nose that contains some specialized cells. Now, these cells have little molecular receptors where odors can bind to them, like you see in the bottom right-hand corner. And when that happens, it can trigger the cell to send a signal to the brain. And there's a special part of the brain called the olfactory bulb. So all of this is how it's the same in how olfaction works between, sharks, dogs, humans, and everything in between. Where things get a little bit different with sharks starts with their external anatomy, or the outside of their body. So if we look at their nose from the outside, you can see these 4 pictures here are a shark, 2 sharks actually, a ray and a skate. And rays and skates are close relatives of sharks, so we include them as well. So what you see is they have nostrils similar to us. But what's interesting is that our 2 nostrils connect together to lead to a single central olfactory cavity, whereas with them, their left and right nostrils or sides of their nose are not connected at all. So they have 2 separate olfactory cavities that lead to the skin and the specialized cells, 2 separate cells of sets of receptors inside their nose on the left and right sides. Another interesting difference is that to get odors into our nose, we breathe in. We use our respiratory system to pull odors in through our nose and run it past those receptors so that we can sample them. And then we breathe out and it comes back out. So our olfactory system is connected with our respiratory system. That's not always the case for sharks. So you can see this shark down here. You can see the nose on the left and right hand side is not connected to the respiratory system. And for them, the way that they breathe, as you may know, is they pull water into their mouth and push it over the gills, and then the gills are able to pull oxygen out of the water. So the nose and the mouth aren't connected at all in this species. But so the way that they get odors into their nose, in this case, is by swimming. So as they swim, that forces water in through their nose so they can sample odors in their environment. These other species here, and especially species that kind of sit still a bit more, do have a connection between their mouth and nose. And this allows them to pump water and odors through their nose so they can sample odors in their environment. Another neat feature of the shark olfactory system is that each of the openings to the external environment is divided into basically a doorway in and a doorway out. So this facilitates a unidirectional flow of water through their nose. So for us, we breathe in and odors come into our nose, and then we breathe out and odors exit the way they came in. So this is a bit different for sharks and their relatives. And then the other point that I like to make that is the most exciting to me is that there's a lot of variation from species to species. And just taking a quick look at these 4, you can really see how different they are in terms of the way their noses are set up. So that's something that I've studied for quite a while because I want to know why. If we take a look at the inside of their nose, it's pretty neat. So I mentioned that water carrying odors will come in the nostril and enter an olfactory cavity, just like us. Well, inside our nose, we have a small sheet of tissue on the roof of our nasal cavity with those receptor cells. Sharks have one of these on each side of their head, which we call an olfactory organ. So it's this big, fleshy labyrinth of tissue, and it has a lot of surface area, and there's one on each side of their face, and that's where you find the receptors. So it's a really cool looking structure, and in fact, it's sometimes called an olfactory rosette because it sort of resembles a flower with all these little petals, and those little petals are called lamellae, and I'll talk a little bit more about those in a moment. So what you're looking at here, of course, is a lemon shark face, and you can see inside the nose. But if we take a look at that lemon shark with a top-down view, I want to show you how close that olfactory organ is, I just showed you, to the brain. So just on the other side, on the back side of that organ, is this pink piece of tissue, which is a part of the brain. It's called the olfactory bulb. And that's where the specialized cells that detect the odors are going to send their signal. It's the first place that odor information goes in the brain. And then eventually, this animal will be able to respond to odors in its environment and decide, oh, that's a food odor. I need to swim after it and actually respond with behavior. So if we take a look again at a brain here, this is an Atlantic stingray brain. You can see those olfactory organs up front. This brain has been dissected out of the specimen. And then behind it, this white tissue is that piece of the brain I just mentioned, the olfactory bulb. And then here's the olfactory tract and the rest of the brain, the cerebellum here. So let's take a look at a cartoon view of the nose portion. So there's the olfactory organ again, and the olfactory bulb of the brain right near it. And then there are those little petals or plates called lamellae. Now I want you to see how odors actually stimulate this tissue. So we're gonna zoom in on one of those lamellae, and you'll see this is where we find those specialized cells. They're called olfactory receptor cells or neurons because they are primary sensory neurons that respond to chemical stimuli. And then if we zoom in on these tiny projections, they're little cilia and microvilli, that's where you find the actual molecular receptors that bind to the odor molecules that are floating around in the water. So once the odor binds to these receptors, it can trigger a response in these cells, and they send their signal straight back to the olfactory bulb of the brain, and then the animal can respond to the odor. The way that we look at the sense of smell, especially the anatomy, often is to dissect the olfactory organ from the shark specimen. So what you see here is the olfactory organ dissected from a bull shark. And it's this big fleshy piece of tissue with all those little lamellae. Another way that we're starting to look at the olfactory system is using what I showed you in that first slide, a micro CT scanner. So the way this works is that we put the specimen in the scanner and it takes a whole bunch of x-rays and then puts them back together into a three-dimensional object. So it's basically a 3D x-ray. So what we're looking at here is a CT scan of a bonnethead shark head. Now bonnetheads are part of the hammerhead family. And you're looking at a top-down view of this bonnethead shark. So up here at the top of your screen, you're seeing kind of the front edge of the head of the shark. The eyes will be on either side, and back here toward the bottom of your screen is where the body of the shark is. Now, once I play this video, you're gonna see we're gonna slice down from top to bottom through the head so you can see the insides of the head of this shark. So we'll start slicing away the top of the tissue. I'm going to pause it here so you can see some of the features that I'm noticing. So right here is the central portion of the brain. You can see one of the eyes starting to appear over here, which is pretty cool. And now you can see on this side of the brain, there's a structure kind of stretching off to the right that's called the olfactory tract, and it's leading to this structure back here called the olfactory bulb that I mentioned before. And here is that beautiful olfactory organ. Now you can see it's missing on the left side because one of our graduate students has dissected it away. But this CT scan allows us to look at the olfactory organ kind of in place as it exists in the shark head, which is a lot more helpful. So based on what I told you before, you can imagine odors floating around in the water, and it comes in that in-current nares, or that doorway into the olfactory capsule. And it floats through this space and interacts with all these lamellae and all the receptor cells on the lamellae, and can trigger a response which will be sent back to the brain. And then that water and odor will flow out the excurrent nares. So you can see some of that as it actually exists inside the head, which is pretty neat. Okay, so when we do dissect out the olfactory organ, we often like to compare how the organs look and how they're shaped and what the structure is like from species to species. So I showed you the bull shark olfactory organ before. Down below, you can see the olfactory organ from the great hammerhead shark. So immediately, looking at this, you can notice a bunch of differences, not only in the color of the organ, yes, but what's more interesting to me is the shape. So the great hammerhead shark olfactory organ is really long and skinny compared to the bull shark and really most other sharks. Hammerheads, as you know, have that weird head shape that's flat and elongated, and that causes their olfactory organ to have a similar appearance. So this is really going to influence how water and odors flow through this structure. So I'm curious about the shape, and that's something that I investigate. The way that we look at the shape of the olfactory organ to get some meaningful data is by taking a bunch of measurements. So we dissect the organ from the shark specimen, we put it under the microscope and take some really nice high-resolution photos. Then we look at those photos of the olfactory organ, and we can do things like count the number of lamellae and measure the distance between the lamellae and how thick the lamellae are. And we can also measure the width of this little structure, which is called the raphe. We also can take a look at the whole organ and measure the length and the width. And actually, we divide the length by the width to calculate a metric called fineness ratio. And that's something that we compare species to species. So you can imagine back to that hammerhead shark olfactory organ. It's very long and not very wide. So that fineness ratio is going to be much greater than you would see for this bull shark. So some of the things we find kind of back up our anecdotal observations that the olfactory anatomy is really different from species to species. So even when we just look at the number of lamellae and we compare 5 different species, we've got 2 shark species, the bonnethead shark and the lemon shark, 2 ray species and a skate. You can see when we look at the number of lamellae that the bonnethead shark has over 60 lamellae in its olfactory organ, whereas the clear-nosed skate has under 30. So a very different number. And you can see just visually, they look very different. When we look at that fineness ratio, which is the length of the organ divided by the width, you can see exactly what we observed, that hammerhead sharks have a significantly greater mean fineness ratio compared to other non-hammerhead sharks and also the batoids. So when we take together all of the observations and measurements of the anatomy of the olfactory organ, what we're finding are some trends that you kind of have 2 ends of the spectrum when it comes to the olfactory organ. So if we take the great hammerhead shark as kind of one end of the spectrum, you have a really large fineness ratio, very long and skinny, a lot of very thin lamellae that are tightly packed. The spacing between them is very thin. And then other species have a smaller fineness ratio with fewer lamellae that are thicker and have a greater spacing between them. Why do we care about the anatomy though? It's because it's going to impact the flow of water and odors through the nose and the ability of these sharks to detect odors. So the way that we study this, and we're just beginning on this set of studies, is to model the shark olfactory organ. So to create a model of a shark nose, we start by looking at a simplified diagram like this, which shows you the olfactory organ, and all of these lines represent those lamellae I mentioned before. And because the organ is bilaterally symmetric, we can just look at half of it and take that shape, and because it's a model, we can kind of simplify it. And here's our model, a CAD model of a shark nose. Okay, so what we can do from here is we can actually build this model. What we did is we laser cut clear acrylic in a really cool lab at our school called the Kane Institute for Advanced Technologies. And this is what these models look like. So these are just our prototypes, but you can see we can vary these models in really cool ways, like make a really long one like the hammerhead shark nose, or a shorter one like a bull shark. The other thing we can do is we can change the number and spacing of those lamellae. And what we do with these models is we put it in this tank over here, which is a flow tank. So this tank is lit up bright green. We put the model in there, and then we push water through this structure. Now in this water, we put a bunch of little particles so we can actually see the way that the water is moving. We shine a laser on those particles and record high-speed video so we can do some analysis of the way, the direction, and the velocity of the particles. And this technique is called PIV, or particle image velocimetry. So the neat part is when we alter our model to make it longer or change the number and spacing of those plates, we can look at the effect experimentally of the flow through the olfactory organ. So here's one of our videos. You can see the little particles kind of moving around and the water flowing through our shark nose model. So there are areas of fast flow, like here. There are areas of slow flow, and there are kind of recirculating areas. So we're starting to build a model of the shark nose so that we can understand the impact of anatomy on flow of water and odors through the nose. The importance of flow through the nose is that that's how odors are going to move through and bind to the receptors. And that's going to affect their ability to actually smell stuff. So this makes us wonder, how does flow through the nose affect function, or their ability to actually detect odors? So first I'm going to give you just a quick background on how we measure this. To record the function of the olfactory system, or the physiology, we use a really cool technique called the EOG, or the electro-olfactogram. This allows researchers to do live recordings of the shark nose and stimulate it with different odors at whatever concentrations we choose, and ask questions like, can a shark smell this particular odor? Or which odors stimulate them the most and the least? Or how sensitive is a shark to an odor? So that's going to get at our question from the beginning. How far away can they actually detect an odor? This is what this technique looks like, which is a little complicated looking. For now, you can ignore the electronics over to the right and take a look at the shark, which is restrained belly side up inside of a tank. And the important stuff in this experiment is happening up at the head. So if you look at this inset in the top left-hand corner of your screen, you'll see there's the eye of the shark, the mouth, and the nose. So we have two tubes going into the nose. This one called ODP is the odor delivery pipette, and it delivers whatever odors that we choose into the nose of the shark. This other tube called the active electrode will record the response of the shark to the odors. And there's a reference electrode over here positioned on the skin. The signals from the two electrodes then go back to all of our electronic equipment to amplify, filter, and digitize the signal, and then I see a signal on my computer screen. So basically, I can see what's happening in real time inside the shark nose by just looking at my computer screen. So we'll see a trace which represents the voltage inside of the nose, and then when I stimulate the shark's nose with an odor, the trace will go down because there's a net negative charge that the electrode is picking up. As the odor washes away and out of the nose, then the trace will come back up to baseline. So in this way, I can test the response of the shark to lots of different odors and see how large the responses are. So here you can see some of the EOG responses of a bonnethead shark to a really stimulatory odor called alanine. So we deliver this odor at lower and lower and lower concentrations to see how low they can go. And that's really how you calculate the sensitivity of a shark to an odor. So you can see as the concentration gets more and more diluted, then the response actually gets smaller and smaller and smaller until right around this range, you can almost not even tell the difference between the response to alanine at this concentration and the response to just putting seawater in their nose. We call that a control. So it's at this range where we can calculate their sensitivity number, or the lowest concentration they're able to detect. The other thing we can do is compare their responses to a lot of different odors to see which ones stimulate them the most. So what you're seeing here are the responses of the lemon shark, Negaprion brevirostris, to 20 different odors, which we see along the x-axis. And the size of their response is on the y-axis. So you can see odors like phenylalanine or alanine or serine are highly stimulatory to this shark, whereas glycine and valine are not quite so stimulatory. So that gives us a view of what stimulates them the most. So here's what we know so far from these experiments, is that sharks use their sense of smell to detect food, which is no surprise, but that's critical for their survival. They also probably use olfaction for so much more. So we think that they also use their sense of smell to detect mates, to detect predators, and maybe even also for homing and navigation. So it's really important for their lives and for their survival. What we know from these experiments, we've tried their sensitivity to a couple of different types of odors. So we know that they're sensitive to amino acids, which are the building blocks of proteins, and bile salts. And in terms of their sensitivity, they can smell all the way down to 1 part per billion, which is down in the nanomolar range if you're familiar with chemical concentrations. So that's really, really sensitive. It's kind of hard to conceptualize what 1 part of odor looks like in a billion parts of water. So that's why on these shark documentaries, you hear things like they can smell a drop of blood a mile away or something like that, because it's easier for us to kind of wrap our mind around. So to get to the question, can a shark smell a drop of blood a mile away? So I'm going to break down this shark fact that we hear on shows a lot of times into 2 parts. The drop of blood part, I'm going to get to in a moment. So can they smell odors a mile away? So it's actually not a great way to set up the shark fact, because if we drop an odor a mile away from a shark in the ocean, the way that that odor moves around is really variable. Is the water moving? Is it caught in a current? Is the water totally stagnant? And then that odor has to diffuse out evenly throughout, that mile. water moves in the ocean. So we can't tell if they can detect something a mile away necessarily. That's not a good experimental setup. So if we take the other version of the shark fact, can they smell a drop of blood in an Olympic-sized swimming pool? Well, that's a volume of water that we know. So I can Google how much water is in that size swimming pool and ask if we take an odor, a drop of an odor, and it diffuses out evenly through that volume of water, can sharks detect that low of a concentration? On to the other part of this shark fact, sharks detecting blood. So sharks aren't interested in human blood. As you may know, if you're a shark lover like me, you know that we're not their prey. So they're not particularly interested in human blood. So that shark fact is actually kind of silly. So whenever you hear it on shows, you don't need to believe it. But just to kind of entertain the idea, when we use the right type of odor that's actually relevant to the life of a shark, sharks can smell about 1 drop of odor in more like a backyard-sized swimming pool, not quite an Olympic-sized swimming pool. That's still incredibly sensitive. But you know what? There are a lot of other studies on the sense of smell of other fishes, and it turns out that sharks are about as sensitive to odors as other fish in the ocean. So maybe they're not the swimming noses or the super smellers that we kind of make them out to be, but really they're fish and they're well attuned to their environment so that they can survive and get food. So I hope that you've learned a little bit about the way that shark noses look and how they work and some of the recent data that we've collected. I really appreciate you listening to my talk, and I'd be happy to answer any questions you have. So thank you.
Well, Dr. Meredith, thank you so much. This was fascinating and so interesting to learn all about this. We have lots of questions that people want to ask you. And so one that I'd love to just start with is, how did you get involved in shark biology and in studying olfaction in the first place? What inspired that?
Yeah, so I didn't grow up by the ocean. I grew up in Ohio, and actually I fell in love with the ocean because occasionally we would vacation in South Carolina, and I would find all the weird little creatures on the beach. So I started getting fascinated in marine biology and kind of set my path from there. And you know, you combine that with watching Shark Week growing up, and I was in love with sharks and couldn't wait to work with them.
Fantastic. All right, so we also have lots of questions about shark anatomy and shark senses. One person has just asked in the text chat, and this is a really interesting question: does the temperature of the water affect how those particles travel through the shark's nose, and as a result impact how far away they can smell those odors?
That's an interesting question, and I'll tell you the short answer is we don't know. So nobody's actually looked at that, and I think that's kind of what drew me to researching their sense of smell. Is I couldn't believe that as recent as a handful of years ago, we didn't even know really basic stuff like how sensitive are they to prey-related odors. So we're missing a ton of basic knowledge about how their sense of smell works. Probably temperature is going to play a role, but we really just haven't tested that yet.
Super interesting. Lots more to discover for those aspiring marine biologists that are joining us on the webinar today. Speaking of which, from a research perspective, thinking about odors in the water, how do we as human scientists know what the sharks are smelling, know how they're smelling it? I mean, can you give us a little more information about how you're actually studying that in the water?
Right, so the olfactory physiology experiments we do actually in a lab setup. So we collect the animals from the wild, just a few individuals. We bring them into the lab and make sure they're acclimated, and then we take one animal at a time and we test their olfactory physiology. There are some experiments looking at their behavior and related to their sense of smell. So free swimming sharks in a tank or out in the wild and how they respond to odors. Those studies are really, really hard to do. But there are some studies out there that suggest that sharks will not only use their sense of smell, but they're using all of their senses together at the same time, of course, to kind of localize prey very quickly. And they need those senses working together in concert to localize prey as quickly as possible.
Very cool. Excellent. All right, so we have a couple people who are wondering about how sharks' sense of smell compares to land animals, whether that's land animals that have a really sensitive sense of smell, or maybe even how they compare to humans' ability to smell things.
Right. Yeah. So this is an interesting question. And I think our understanding of this is changing rapidly over time. There are a lot of people that study the sense of smell of mammals and especially humans. And we use mice and rats as model organisms for that. So we know quite a bit about their sensitivity. People have studied other mammals as well, like dogs, because they're famous for their sense of smell. The anatomy is really different from group to group and even within groups, as you saw with sharks. But it turns out that sensitivity wise, we're not all that different. So we sort of have these ideas of who the super smellers are and aren't, and that we don't rely on our sense of smell, so we're not that great. But the key is to choose the right odor stimulus. So, you know, if you are waving a steak or spraying some steak odor in front of a panda bear, they're not going to really care because they're not, you know, a carnivorous predator necessarily. So you have to really choose the right odor to stimulate the system of the animal that you're looking at. So it's hard to compare because we're going to use different odors for different animals, but we have a similar range of sensitivities. It's what's coming out.
And I
Interesting.
have a quick question, Trisha. I do remember being in the lab with you at one time, and I think I remember you taking your own blood for one of your EOG experiments. I remember thinking how crazy you were. Can you comment on that?
Yes, I think I was plagued by this thing that we hear in shark documentaries all the time of sharks smelling human blood, and I just wanted to try it. And it wasn't a very scientific experiment, and in fact, we barely saw any response at all. But I thought, you know, if I could just squeeze a little bit of my blood into this experimental apparatus, you know, not my finest moment.
I remember. Well, I have one other follow-up question that I think is interesting, and hopefully I'm not going in front of a student who wants to know this, but I'm wondering, as you start to study, you know, the morphology of the olfactory rosette and all of that, are you starting to think about how that size and structure might correlate to the animal's ecology?
Yeah, that's a great question. So there's a great scientist, Vera Schlussel, who did a really comprehensive study not too long ago looking at how the shape of the nose and all of those morphological calculations that we make, how that relates to either ecology. So are they benthic? Are they benthopelagic? Are they pelagic? What kind of prey are they eating? Are they eating crustaceans? Are they eating fish? And she also looked related to phylogeny. So is your nose structure more likely to be caused by who you're related to or what your lifestyle looks like? And she had some evidence to suggest that there was some differences between benthic and non-benthic sharks, but it's really hard to tease apart those differences because species to species differences are so great.
Interesting. Thank you.
Yes.
Super interesting. Absolutely. Okay, so we have a question from a student actually that's been tuning in. If sharks can smell something that's maybe just above the water, so not necessarily down in the water column with the shark, but in the air around that part of the water that they're swimming in, do we have any research on that?
That's a cool question. So, you know, that makes me think of if you've got like a whale carcass and some of that odor or stench is up in the air, how is the shark able to find that? Because we see these amazing videos of white sharks finding these whale carcasses and, you know, feeding in a frenzy. So how are they finding that? What has to happen, you know, you can't really get around it, is those odors are little chemicals. They're little molecules floating around in the air, and some of them will get into the water and dissolve into the water. And that molecule of that chemical has to get through the water and physically enter the shark's nose in order for them to smell it. So, you know, if it's in the air, it's probably also in the water. And if that water is moving, or if the shark is nearby, and we know that sharks are still pretty sensitive to odors, even if they're not more sensitive to other fishes, then the sharks can probably detect that odor.
Super interesting. Thank you. So we have another question. We think about how humans, our sense of smell is really related to our sense of taste, for example. We use our senses in combination. Is there any evidence that sharks also use their senses in combination? And if so, how do they go about that?
Yeah, absolutely. The short answer is similarly to us. So they're receiving a lot of information about their environment around them from all of their senses all the time. So, if they're very motivated to feed, they're super hungry, then they're going to be picking up on any signals that relate to prey. And the different senses will operate kind of at different distances from that prey. So from very, very far away, you may be able to hear the sound of a struggling fish through the water, or you may be able to smell some prey that, that the odor chemicals have traveled to your nose. And then as you get closer, you'll be able to see it. So vision is going to kick in. You're going to be able to pick it up with your electrosensory system. So as you're pretty close, then the electrosensory system will kick in, and then eventually taste will kick in as you're taking a bite of that prey. So they're constantly collecting information from their environment as they're looking for prey or mates or trying to avoid predators.
Wow, fascinating and very cool that it does have some similarities between humans and sharks. A somewhat similar question is thinking about how oftentimes as humans grow older, their senses will change. And so people are wondering if throughout a shark's life cycle, will the senses that they rely on change throughout the course of their life?
Yeah, another fantastic question, and I think a future PhD dissertation for the question asker, because again, we don't know the answer. So, it's a shame that we're missing so much basic information about this critical sense for these species, but that's a whole area of study. How does the sense of smell change throughout what we say ontogeny, throughout the growth and life of the organism? Probably it does. It would make sense if it does, because the prey that they consume will change over their lifetime, and it's going to be a critical sense to find and localize that prey. The predators that they're vulnerable to will change throughout their lifetime. And we know this from the other fish literature. So not necessarily sharks, but in other fishes, their sensitivity to their conspecifics, so especially during mating time, to be able to smell a female is really important so that you can perpetuate the species, obviously. So that sensitivity is going to change from when you're a young fish to one who's able to mate. So probably it would make sense, but somebody has to investigate it.
Interesting. Okay, thank you. And again, lots of potential questions for the future marine biologists in the audience to think about. Fantastic. So one person is wondering about, we talked about those lamellae, and whether or not the number that they have, is it all related to what that shark is going to eat? So do we see more of those if a shark's going to eat bigger prey or things like that?
Yeah, so that was one of our initial questions. What's up with the number of lamellae differences? And does it relate to ecology, to prey, to phylogeny? We're always trying to figure out why, or even sensitivity. So that's what I was thinking. Maybe the more lamellae, the more surface area for receptor cells, the more sensitive you are to odors. But really, we're not finding those relationships. So I'm still on the hunt to figure out why there are so many differences in shark noses. My guess is that it has something to do with optimizing the flow of odors through the nose, which is why we're sort of going that path with our research, but I really don't know the answer yet.
Super interesting. Thank you. So we've got some people wondering about, as we know, sharks aren't really interested in humans as their prey, but there are these occasional stories we hear in the headlines of maybe something happening. And so someone's wondering, can sharks really smell the difference between, say, a drop of human blood in the water and a drop of prey blood that they actually want to go after in the water? Can they really sense those differences?
Yeah, so I think the answer is probably yes. Of course, we're not testing in a real way, despite what Dr. Mickey just told you, their sensitivity to human blood. But blood is a mixture of lots of different molecules and chemicals. Our blood is different from the blood of a fish, for example, or, let's say there's an injured fish and some of the tissue, the chemicals in the tissue are kind of an odor cue for a shark. So all of those are going to be comprised of different sets of molecules. different chemicals at different concentrations and different mixtures. So yeah, their sharks' noses are really attuned to their prey and to the odors that are relevant to their lives. So they're not going to be as sensitive. It doesn't make sense to kind of build your whole sensory system around odors that don't make a difference in your life. So they're really much more sensitive at detecting those particular odors that are relevant to their lives.
Interesting. Very cool.
Yeah.
Oh, yeah.
I have a question for you. So, Trisha, you know, it's interesting to think about, and you just touched on it a second ago, pheromones. And, you know, it's interesting to me to think about what you just said, you know, receptors that are ready to bind a particular molecule, a pheromone that might be released. What do you know about sharks' ability to detect pheromones? And what do we know? What's the state of the state with understanding what pheromones are shed by sharks? What, you know, what's the knowledge that we have on that right now? Do you know?
Yeah. So as I think you know, we know barely anything about how sharks use pheromones, which is crazy to me because of course they probably use pheromones. Other fishes do. So these are, you know, conspecific cues. We see some behaviors that make us think that they probably use pheromones. So we'll see sharks swimming where, you know, if the female is emitting some pheromones from her cloaca, we see the male with his nose close to her vent, and that would suggest that he's picking up on those odor cues and maybe sensing that she is ready for mating. But we haven't really tested it. It's tricky to test. Sharks are hard to work with in general, which is why a lot of this basic research hasn't been done yet. But when we look at other non-shark fishes, we know that they do use pheromones for lots of different things. And so it's really likely that sharks do as well. We just need to test it.
And then one last thing, what about the sense of taste? So you talked about, you know, multi-sensory integration. What about the sense of taste? Is anybody working on that?
Yeah, barely anybody. I know, I'm sad that I can't answer any of these amazing questions. They're questions that I also have, but somebody needs to study the shark sense of taste. We need another one of you students out there to do that for your dissertation.
Absolutely.
And someone, Dr. Meredith, someone's wondering what sharks are next on your list to model? Is there another shark that you're like really curious to learn about?
You know, the cool thing about the modeling is not only are we going to model the hammerhead shark nose and then the non-hammerhead shark nose, which is not quite so long, but then we can model sharks that don't even exist. So we can make that shark nose model any shape, any fineness ratio we want. We can put as many lamellae in as we want, even ones that don't exist in nature. So that's the really super cool part about physical models, is you can test things that you see in nature, and then you can take it a step further and try out combinations that don't even exist and see the impact on the flow.
Great.
So I'm looking forward to doing that.
Yeah, I have one more question. So and I know you talked about this, but it begs the question anyway. Hammerhead heads, huge, you know, disparity in the length of the lamellae. And the question is, are they more sensitive? Is there anecdotal evidence that they show up on the scene faster than other sharks? Is there some kind of way they can sense odors with these narrow space so far apart? Can they see gradients or sense gradients of odor, things like that?
Yeah, hammerheads are a cool case to consider because of that weird head shape and how it impacts all their sensory structures. So, like I mentioned before, our nostrils are really close together and they lead to just a single cavity. With sharks, their nostrils, their noses, left and right noses, are separated. With hammerheads, their two olfactory capsules are way off on the ends of their head. So even further physical spacing. So that makes us think that as they swim through a gradient of odor, they might be able to like instantaneously detect concentration differences between the left and right sides of their head at any given moment, which is really cool. And there are some experiments showing that. So they're able to take hammerhead sharks, I believe it was a bonnethead shark, in captivity and place little kind of tubes inside of their noses and deliver odors on left and right sides to see how it influences their turning and swimming. So there is some evidence to suggest that their weird head shape is influencing their behavior related to smell. And a cool study by Dr. Kajiura showed that they may be even a little bit more sensitive to odors. So their sensitivity to a couple of particular odors was down in the range of 10 to the negative 11th molar, which is a little bit more sensitive than what we were recording in most of the other species. So we're still figuring out what's going on with that weird hammerhead head shape, and if it confers some advantage to their sense of smell, but it's looking like maybe it does.
That's so cool. This next question this next question I think is really appropriate for Earth Day. We have a couple of people who are wondering if a shark's living close to a coastline that might be more polluted with odors from construction or sewage or just poor coastal health. Will that cause migration? Will that cause them to leave that habitat because of those smells?
That's a great question. The short answer is we don't have a direct, study that answers that. But what we do know is that pollutants can really mess up their sense of smell. So there was a pretty cool study not too long ago. Eloise Cave did this study looking at the Deepwater Horizon oil spill and if sharks swimming around in that pollutant had any impact on their ability to detect odors, and it really did impair their sense of smell. So there are some studies, some physiology studies using the electro-olfactogram that I showed you that sort of demonstrate a similar concept that pollutants can really mess up their sense of smell. So it's going to get in their way of detecting prey, which is, really a big problem because they're going to rely on that for food. So whether or not they're going to try to avoid those areas, I'm not sure, but they certainly would need to, or it's going to impact their survival.
Great, thank you. So we'll have time for just one more question, and I love this question. We sometimes think about how humans, right, if we get a little bit of black pepper near our nose, we'll sneeze. Can sharks sneeze underwater?
You know what? I'm not sure. I've never seen a shark sneeze. Maybe Dr. Mickey can chime in, but
never
to be determined.
I've seen a sea turtle yawn, but I don't think I've ever seen a shark sneeze.
All right, more questions for future marine biologists.
Yes.
Well, Dr. Meredith, thank you so much for your time today and for all the incredible insight you've shared with us. This is so fascinating. I want to let everyone in the audience know that this has been recorded and will be available up on YouTube by the end of the day, so do feel free to share what you've learned here with others. We'll also be sending out a fact sheet and some activities to folks who signed up for the webinar with their email addresses. This will allow you to maybe do some at-home experiments with your own olfaction and to continue learning about shark sensory systems. I also want to let you know, if you enjoyed this, we'll be doing another Zoom live stream as part of Ocean First Institute tomorrow. Dr. Mickey and I will be back with some other special guests talking about celebrating our blue planet since Earth Week. And finally, for those who are interested, our resource page on our website, oceanfirstinstitute.org, has lots of activities and fun facts that you can do to celebrate Earth Day. So with that taken care of, I'm going to turn it back over to Dr. Meredith and Dr. Mickey for some final words of insight before we head out today.
Great. Well, I would just like to say, Dr. Meredith, it has been a pleasure to see you again, and I thank you for your time today coming and sharing all of your knowledge and your research with us. It's extraordinary, and to especially do this on Earth Day, it just shows us how important learning basic things about these amazing animals is and how much we really don't know. So I'm going to turn it over to you for goodbyes, but thank you, Dr. Meredith, for coming today and for sharing your story with us.
Yeah, I really appreciate you guys having me. I had a great time sharing my passion of shark noses with you. If you have any other questions, I'm happy to answer any follow-ups. I'll be on social media, so feel free to get in contact. Thank you so much.
All right. Thank you, everybody.
Thanks,
Happy
everyone.
birthday. Bye.